Alright, so let's start talking about the basics of tissue culture. But before we start talking about the basics of tissue culture, let's go over what the study of cell biology is, because this is Cell Biology Lab, and we're going to be talking about techniques that are used by cell biologists to study the cell. But what are they studying? So cell biology is a branch of biology which is dealing with eukaryotic cells in multicellular organisms. There are different branches of biology that deal with eukaryotic cells that are unicellular, such as mycology, and the study of prokaryotic cells, for example, bacteria, like microbiology. Cell biology deals with eukaryotic cells and multicellular organisms. So why is this important? Because different cells in your body are independent agents that work in concert with each other. So if I had two cells, for example, here's cell A, and here's cell B. And let's say these are different cell types. We know that these cells are genotypically equivalent. They're both somatic cells. These are not germ cell lines. They're not gametes. So they're genetically equivalent. So genotypes equal each other. So genotype A equals to B. They have the same chromosomes, the same genes, etc. Phenotypically, they're going to be different. Now, these cells, even though they're genetically equivalent and they're independent agents, within a tissue, within the organism, they're working in concert with each other. And the whole study of cell biology is trying to figure out how does one cell communicate with another, for example, A communicating with B, to get B to have a specific response. How do you get this response? How do they communicate? How does this response happen? For example, your nerve cell telling your muscle cell to contract. How does that happen? That's actually a topic that we're going to delve into in the lecture, but that's just an example. So the whole study of what we're going to be talking about in lab, at least this semester when we go over different tools that cell biologists use, is to sort of understand how this process of trying to figure out what is going on happens. So the culturing of these cells, removing the cells from the organism and putting them into a petri dish, that's called tissue culture. Right? So tissue culture is simply growing cells in a petri dish, or maintaining cells. So this is typically called the growing of cells or maintaining cells outside of the organism, an in vitro environment. And when the cells are in the organism, it's called an in vivo environment. So in vivo. So let's go back to our study sheet. So the basics of tissue culture. Advantages of tissue culture. Control of the environment. Control of the physical parameters, which include temperature, pressure, and viscosity of the medium. So basically here, if you're growing cells in a petri dish or some other vessel, you can control the environment those cells are in, the physical environment, very well. You can increase the temperature, lower the temperature, you can add more pressure if you wish, or you can change the viscosity of the medium that they're in, make it more viscous or make it less viscous. And these are all physical parameters that can be controlled very easily. Now we can also control the chemical parameters very easily. We can control the pH, availability of reactants, which can include monomers and polymers and biomolecules. So this would include stuff like amino acids and nucleotides, sugars, etc., right? Or any other cofactor that you wish to control its concentration in the medium. Enzymes. So you can control the level of enzymes within, and enzyme inhibitors. You can add enzyme inhibitors. So these are all chemical parameters, because pH is a chemical parameter, you're controlling for the concentration of H+. Availability of reactants, well, with or without the reactants, then the reaction cannot happen. Enzymes, well, if the enzyme is not there, even if you have the reactants, the reaction cannot happen. And inhibitors, if you inhibit the enzyme, then the reaction of the enzyme is controlling will not happen. So these would be all under chemical parameters. Of course, there's more to these physical and chemical parameters. I'm just listing a few examples. So the next big thing is homogeneous constitution. A homogeneous constitution does not mean that every cell in a culture are equivalent genetically or phenotypically. So this is kind of a big one that we're going to spend a little bit of time on. So when we're looking at cells in the body, so let's say we're looking at liver cells. Let's just go with hepatocytes, liver cells, okay? So in the liver, you have these cells that basically do the work of the liver. They're called hepatocytes. There are other cell types within the liver. There's resident immune cells, other cell types that are there to maintain and help the organ do its work. But the main work of the liver is done by hepatocytes. So these liver cells, you can imagine that if you have them in epithelial tissue of the liver, they're sort of next to each other. Right, and you have a basement membrane here, which is a bunch of proteins, and they have proteins that bind to this basement membrane, keeping these cells tethered to the basement membrane so they don't just fly off. And here are the nuclei just to make it look like cells, right? Now, each one of these cells, and we'll label them A, B, C, D, and you can go in either direction. There's going to be more of them, right? Each of these cells is in a slightly different environment. They're genotypically equivalent in this case, right? But they're in a slightly different environment, so their phenotypes will be slightly different. So a hepatocyte in one part of the liver might be slightly different in its phenotype than a cell in a different part of the liver, although they're the same cell type. So just a slight phenotypic difference. Now, if I take these cells from this in vivo environment and I put them in an in vitro environment in a petri dish, so here's my petri dish, and I put these cells in there, they will bind to the bottom of the plate and form a monolayer. And those same proteins that are binding them to the basement membrane, just to make sure that we know this is the basement membrane, those same proteins will bind them to the bottom of the dish. Now, we said that these cells are going to be slightly phenotypically different from each other. So you'll have different populations that are slightly phenotypically different from each other. So let's say that, for example, we have one type of phenotype that is A, and we'll have a few A's. And we have another one that is B. So let's say B, and a few C's, and a couple of D's. These are still hepatocytes, liver cells, but they're slightly phenotypically different from each other. So when I grow these in culture, they will replicate. So one cell will give 2 cells, 2 cells will give 4, but in this case, what do I have a ratio? I have a ratio of 1, 2, so I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 cells here, So 1, 2, 3, 4 A's, 2 B's, and C's I have 1, 2, 3, 4, D's 1, 2, 2. So that gives me 12, right? Now, this ratio of 1/3 A, 1/6 B, 1/3 C, and 1/6 D that mixture, those proportions will stay the same as these cells replicate. And that's what a homogeneous constitution means. It means that although they're not exactly the same, they're pretty close to being the same, but they're not exactly the same. I will maintain this proportion throughout my experiments. So when these cells grow and I go from 1 plate to 2 or 3 or 4 plates, I am pretty sure, almost 100%, that I will have the same proportion in those other plates. And that's what homogeneous constitution means. Now, the other issue is that we said they could be genotypically different. Now, what does that mean? Well, if I take cells directly from the tissue, so I go directly from tissue into an in vitro environment, petri dish. These cells that come directly from the tissue are called primary cells. So these are primary cells. So we'll just say 1', right? Now, primary cells will divide a few times and then they will die. So they will divide, 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 division, mitosis, right? And then they will die. And this is not very useful because if you cannot maintain the cells indefinitely, you will have to keep going back to the organism for more cells, and that's not a viable option. So when we take cells from the tissue, there's a couple of things you can do to have cells that will not die, that they can keep dividing and being maintained in vitro for an indefinite period of time. One of them is that you take cancer cells. So you take cancer cells from tissue, so cancer cells from tissue, and maintain them in vitro. And they can divide and divide and divide and divide, and they will keep living. So you have an indefinite supply of cells because they will keep dividing and they will stay alive. So if I need 2 plates of cells or 3 or 4 plates of cells, I let the first plate divide, then I split them into the 4 different plates, and they will again divide and fill up that plate. The other thing you can do is you take normal cells from tissue, so normal cells from tissue, and you make them cancerous. This is a process called transformation. This is in vitro. And now they will divide indefinitely and can be maintained indefinitely. Now there's a problem. Once a cell becomes cancerous, it gets this property called genetic instability. So cancer cells are genetically unstable. What does that mean? That means upon many rounds of division, at each division, the genotype will change, sometimes very slightly, but sometimes dramatically. So this genetic instability will lead to having cell types that are not only phenotypically different, but they can be genetically different from each other. But you can be sure that, just like the example above, if the cells are maintained correctly, you will have different populations at different ratios. And you can be sure that that will not change over a long period of time. We will be talking about when you can find out if something has changed dramatically and how to be aware of that. But for now, what we mean by homogeneous constitutions, that even if I have a cell line that has been transformed or it's from cancer tissue, it's a cancer cell line, that within a period of time, after each cell division and passage of these cells, that I will have the same constitution, the same ratio of different populations of the cell within that culture. Now, why is this important? Why is homogeneous constitution important? Well, if I'm doing an experiment, studying some drug, let's say, so drug A, I am throwing this drug onto the cells and I'm measuring something. So, measuring. A parameter. Now, if I do this experiment later, or while I'm doing this experiment, my conditions change, then I could have different outcomes. So if I have condition condition A, condition B, condition C, I might have different outcomes. Right? Now, in some cases, it could be part of the experimental design to change the conditions because you're measuring something very specific and you want to see how changing the conditions affects that measurement. But the one thing that you might not want to change is the cell population and how your cells behave. So you want your cells to be the same, behaving the same way, even though you're changing some external or internal conditions. So essentially, that is one condition that you want to keep constant. The cells you're doing the experiment on. Because if that is changing, and it's changing in a way that is not under your control, then any of these measurements will be questionable and cannot be reproduced. Okay guys, we'll stop here and continue later.